Alrighty. Good afternoon, everyone, and welcome to the first of our 4 virtual field days that I have been working on for the Research Center here in Monmouth. Since I'm the speaker presenter today, I'm not going to go into an introduction of myself because you'll get that in the video. But I am Chelsea Harbach, and I'm very happy to have you all here with me today. Before we jump into the video, I know it looks a little scary seeing a PowerPoint up here. I promise you it'll be more engaging than just a PowerPoint, which reminds me I was actually going to turn on my video so you can see my face if that interests you. So here I am. And so before we dive into the video, we have just a few housekeeping items here. You can submit questions throughout the virtual field day in the chat box. Or after the presentation, you're welcome to unmute yourself to ask questions. If you are struggling to find the chat box on Zoom, you can look for this these ... symbols and click on that, and you'll see the option to open up the chat box. This field day is brought to you by our Research Center staff and crew. There's Alan Alan Parrish, who actually directs all of our crop sciences research centers for the University of Illinois. Greg Steckel, who's our on-site research agronomist, as well as Marty Johnson, who's our senior research specialist and makes a lot of the stuff happen here on the farm, as well as myself. And a big thank you to the folks in the area, the different companies that kind of help keep our research farm afloat. We get donations from all of these people: seed, chemical, fertilizer, what have you, and all of this stuff really contributes to keeping our research farm going. So you're here for today's topic on soybean cyst nematode. Here's a picture of me, Chelsea Harbach, and I'm going to go ahead and get the video started so you can get my introduction. So here we go.
Hello, I'm Chelsea Harbach. I'm a commercial agriculture extension educator for the University of Illinois. I am stationed at the Research Center here in Monmouth at the Northwestern Illinois Agriculture Research and Demonstration Center. I've been in this job for about exactly 1 year. I came here from Iowa State University, where I earned my PhD in plant pathology. I worked with Dr. Greg Tilka and studied the soybean cyst nematode in cover crops. And today I'm actually going to talk to you about soybean cyst nematode. The soybean cyst nematode is a microscopic plant parasitic roundworm. There are, a bunch of different plant parasitic nematodes that can be found in the soil, but this is the plant parasitic nematode that most severely impacts soybean production in the United States. This is actually the top yield suppressing pathogen of soybean in the United States and has been for over a decade. The tricky thing about soybean cyst nematode is that you can have up to 30% yield suppression before you see any above ground symptoms. And in order to confirm that you have soybean cyst nematode, you have to actually dig up the roots and look for the cysts, the adult females on the roots, or you have to collect soil samples and send them off to a lab to have them look in the soil to determine whether or not there are soybean cyst nematode eggs present within the soil. This is a nematode that, unlike some of the other plant parasitic nematodes, forms a really specific and kind of special relationship with the soybean roots. When the second stage juveniles hatch in the soil, they will travel to the soybean root, enter the root, where they will establish a feeding site, and that occurs over time as they exude these specific proteins which manipulate the plant roots to form this kind of specialized feeding site that's called a syncytium. And this specialized feeding site acts as a photosynthetic sink. And that's how the soybean cyst nematode really causes a lot of yield loss, yield suppression, is by having, multiple nematodes in the soil or in the root, enter the roots, and you end up having a pretty large photosynthetic sink that where the photosynthates that are produced in the leaves end up traveling down to help feed the roots, but the roots are being tricked by the nematodes, which have formed these syncytia that are the photosynthetic sinks. So you have photosynthates being diverted from the grain to the plant roots, or from accumulating plant mass to the plant roots. The soybean cyst nematode first appeared in the United States in North Carolina in 1954. And has since spread all the way across the soybean growing region in the United States. It kind of made a big jump to Missouri, and then a jump to south central Minnesota, and has just kind of worked its way into Illinois and Iowa, and has now been found in every single county in both Illinois and Iowa, the top yield or the top soybean producing states in the country. Scientists did identify 7 sources of resistance, plant introductions, the PIs, throughout the soybean germplasm, and these are the plant introductions, the PIs, that are used to determine the genotype of a soybean cyst nematode population in a field. One of those PIs was PI 88788. PI 88788 turned out to be the source of resistance that was most easily integrated into commercial soybean cultivars, because it had the least amount of yield drag when it was introduced into those soybean cultivars. So it was one that was used a lot. You've also seen, you can find some Peking sources of resistance, and those are really the 2 that you can find right now in the market. Since PI 88788 has been the source of resistance that we have been relying on for years, you can have some issues arise as far as SCN resistance to that genotype. Typically, in the past, what has been recommended for managing soybean cyst nematode is using resistance and crop rotation, and even rotating those sources of resistance. So you would plant PI 88788 soybean one year, followed by corn crop the following year, and then maybe a Peking source of resistance, and then corn, and then do it all over again. And the benefit, one of the great things about this nematode is that by simply rotating to a non-host crop like corn, you can see a pretty significant decrease in the population density of soybean cyst nematode. My advisor from Iowa State estimates up to a 50% decrease in the population density just from planting 1 year of corn. However, you don't see that much of a decrease in subsequent years. Now, because this nematode forms a cyst which holds the eggs, these cysts are very durable structures in the soil and can protect the eggs in the soil for a very long time. So you might think, if I just plant continuous corn for a few years, I won't have a soybean cyst nematode problem, but that's not true. Those eggs can remain dormant in the soil for about a decade or more. So really, once you have soybean cyst nematode, there's no getting rid of it. You've got it, and you've got to manage it. You've got to be conscious of it. So as I mentioned, we've really relied on the P.I.88/788 source of resistance to manage soybean cyst nematode for a long, long time. This source of resistance was introduced publicly, so it's not a trademarked trade. It's something that you just get for free with your soybeans. So you might not have been thinking about soybean cyst nematode for a long time because you have this resistance that you're planting and you don't really need to think about it. It's just there. But similar to planting BT corn or using glyphosate on our weeds, if you're using the same source of resistance over and over repeatedly, you're likely going to start selecting for a pest population that can overcome that source of resistance. So if you're planting P.I.88/788 over and over again, you're likely going to develop a SCN population that is resistant to that source of resistance. And really the only way to determine if you've got a resistant population of soybean cyst nematode in the field is to collect soil samples and send them off to a lab and have them run an HG type test. HG stands for Heterodera glycines, which is the Latin name of soybean cyst nematode. And this HG type test will test the soybean cyst nematode population from your soil sample against all the sources of resistance, and will determine how well that population can reproduce on the different sources of resistance. So if you get a HG type test results back and you've got a HG type 2, that means that your soybean cyst nematode population has the ability to reproduce on P.I.88/788 source of resistance. That's what the indicator number 2 means. Now, Nathan Klachevsky has been conducting a survey across the state. He's our field crops pathologist located on campus. He's been conducting this survey for a few years now, where he's asking people to submit samples, soil samples from their soybean fields, collected either in the spring prior to planting or in the fall after harvest. And these soil samples are being tested, being put through that HG type test. And we're seeing some interesting things from that survey thus far that might have some interesting management implications.
I'm going to talk a little bit more before we open it up to questions. So obviously, we're just going to continue talking about soybean cyst nematode. But the first thing I want to do is actually break down this HG type test a little bit further, because to be perfectly honest with you, I mean, maybe, I only have my experience to speak from, but I remember learning about this HG type test in my Intro to Plant Path class in undergrad, and it was pretty confusing for me. So I want to try and break it down a little bit for you so you understand why we're doing these HG type tests. So HG type tests, like I mentioned, are used to determine how well an SCN population can reproduce on the different soybean cyst nematode indicator lines. And these are the indicator lines that are used to breed resistance into our commercial cultivars. And here's the list of indicator lines, and they're assigned numbers here, 1 through 7. So that's how we identify the indicators by their number here. And the first step in an HG type test oftentimes is to increase that SCN population density in the soil from the sample. This is not this step isn't something that we do necessarily every time we run an HD type test. It largely depends on the SCN population density in the soil sample that was submitted to whatever SCN lab you're submitting to. For Illinois, it would be the plant clinic on campus. So those, the SCN population density can be increased by planting an SCN susceptible soybean and let it grow for 30 days. And then the resulting population can be used to plant the HG type test. So the soil from the population increase is separated into these smaller containers. Here, I guess I should point with my arrow, not my hand, is planted into these smaller containers. And then the different indicator lines are placed into these small containers. And then, as well as Lee, which is our susceptible cultivar that we compare all other indicator lines to. So those again grow for 30 days. And 30 days isn't some arbitrary number. It is the amount of time it takes for soybean cyst nematode to undergo an entire life cycle. And after that 30 days, the roots are removed from the soil and the soil is carefully washed away. And then we use a high pressure water spray to dislodge the females from the soybean roots. And we use a sieve like this to catch the females. Now this is a lot of females. Here is hundreds of females. So this is something that you would only see coming off of a soybean cultivar that is susceptible to SCN. But that's what the females look like when you dislodge them from the roots. After all of the females are dislodged from the soybean roots, we can then count them. And then the next step is, well, I mean, the whole process is important, but this is where we actually get the data we need to determine the female index for each indicator line. And we do that by dividing the number of females that were observed on each indicator line by the number of females that were observed on our susceptible check Lee and multiply it by 100. So we're gonna, for example here, if we had 112 females develop on Lee and 25 females develop on PI 88788, for our female index for PI 88788, we would do 25 divided by 112, which we get 0.22, multiply that times 100, and then we get 22%. So if we were going to do this, so this 22% isn't an arbitrary number. We need to compare that against something. And the threshold that we have set for soybean cyst nematode is that if there is a soybean cyst nematode population that has a female index of 10%, meaning that it reproduces, or 10% or greater, meaning it reproduces 10% or more, or at least 10% of the amount of females that you would observe on the susceptible Lee, we're gonna say that that SCN population can overcome that resistance if it has 10% of the females observed on Lee. So to break that down, say we have, here's our indicators as well as Lee, and to make this simple, we're saying that we observed 100 females on Lee. So here the number of females is essentially the female index. So comparing that to our 10%, if we have, we would have a minus for Peking, a plus for PI 88788 at 17%, a minus for number 3, a minus for number 4, a plus for number 5, a minus for number 6, and a plus for number 7. So if we have 2, 5, and 7, our HG type is 257, meaning that this SCN population can overcome the resistance from indicators 2, 5, and 7. The reason this HG type test is important is so we can understand the kind of populations that we have in our fields, and we can better manage those populations, knowing more about the genotype the population. This map here is from a figure from the SCN coalition that shows the percentage of SCN populations in a state or province here with Canada with elevated reproduction on PI 88788. And here you see Illinois, 88%, whenever this survey was done, 88% of the population surveyed could reproduce at an elevated level on PI 88788. But this survey was done some time ago. So as I mentioned, Nathan Kluszewski has been working on a new round of SCN HD type surveys. And he started this in 2018, where they collected 21 samples. And from those 21 samples, we had 2 samples that were HG type 1257 and 19 that were 257. However, all the samples included that number 2, which means that they were all able to effectively reproduce on that PI 88788. And this is from 15 counties. So here are the 15 counties. Obviously, this isn't really a complete survey of the state of Illinois. So the survey was meant to proceed into 2019, and it is worth noting that the best time to sample for soybean cyst nematode are in the spring prior to planting or in the fall after harvest. And if anybody out here was, who's also from the Midwest remembers 2019, or if you've tried to put it in a distant memory because it was so miserable, we'll remember that our spring was terribly wet and our fall was also very wet. So unfortunately, we don't have any soil samples to look at for 2019. However, this survey has progressed into 2020. We've collected a good number of samples this spring. We don't have any results yet from the 2020 survey. As I mentioned earlier, this HG type testing process can take at least 2 months. So we'll hopefully have those results later this year, and also hopefully get some more samples yet this fall. Now, when it comes to managing an SCN population with an HG type 2, where that population can reproduce at an elevated rate on the PI 88788, which dominates the sources of resistance on the soybean market, the biggest thing is continue to be rotation. And I know I mentioned this in the video, but just to reiterate how important it can be to rotate with a non-host crop. Obviously in Illinois, we're not growing soybean on soybean, so that's not generally an issue. But remember that the amount of population decrease you see after one year of corn does not, we don't see those decreases in year 2 or 3 of corn. So it's not only rotating to a non-host crop, but again, rotating those sources of resistance when you're able to. And the other source of resistance you'll be able to find is mostly peaking. There are obviously some seed treatments out there on the market for soybean cyst nematode, but these seed treatments generally aren't going to provide season-long protection, and their efficacy is pretty variable, as well as there's been a lot of talk, and as I mentioned when I was introducing myself, this idea of cover crops impacting soybean cyst nematode population densities. And I encourage you, if you're thinking about using these two methods to try and manage soybean cyst nematode, that you do not solely rely on them for soybean cyst nematode management. The results I saw for my cover crop experiments were not conclusive as far as cover crops impacting soybean cyst nematode population densities, and I've seen really variable results from the seed treatment experiments done at various universities. So again, if you're going to use these two methods, it's going to be important to just use them as a component of your general IPM program to manage SCN that starts with rotation with the non-host crop and rotation of sources of resistance. I want to direct you to a couple of resources here. I did mention the Soybean Cyst Nematode Coalition in one of the pictures I presented. This is a fairly new coalition. I believe it started in 2016. And this website has a lot of resources about SCN, some really nice fact sheets if you want to learn more. I highly recommend checking out their webpage. And then if you're in the state of Illinois, our University of Illinois Plant Clinic does do nematode testing. That includes just soybean cyst nematode counts, as well as the HG type testing. And then we also offer, the Illinois Plant Clinic also offers Illinois SCN testing, where they will screen the soybean cyst nematode populations for just 3 of the indicator lines, Peking, PI 88788, and PI 437654, I think, indicator number 4. Those are the sources of resistance that you will be able to find commercially. So it's an abbreviated test that gives you more producer, gives you information that's more relative to the producers. And they also do corn nematode assays if you want to get your fields checked out for corn nematodes. And with that, I thank you. And now we'll go ahead and open the floor for questions. Again, you're welcome to unmute yourself or submit questions through the chat. If you are interested in submitting samples for this survey this fall, feel free to contact me or Nathan, and we'll coordinate with you on those.
I had a quick question. You mentioned you've done, I don't know if you've done trials on cover crops. I was curious of what cover crops you were looking at.
So I did these trials while I was at Iowa State. I did them, so that was 2015 to 2019. The trials, or the treatments I included in my experiments were, there were a couple different annual ryegrass cultivars, a couple cereal rye cultivars, one tillage radish, one daikon radish cultivar, no, 2 daikon radish cultivars, 2 oilseed radish cultivars, 2 mustard cultivars, and then there were 3 mixes that I included as well. Another question here in the chat: if you are rotating crops, do you draw the test in soybean fields or corn fields? So it kind of depends on your purpose for the test. If you want to soil sample to know how to manage your soybeans in a year following corn, it's perfectly fine to soil sample the cornfield after harvest. Otherwise, when you're soil sampling in before soybean planting or after soybean planting, those are going to be more informational for you. You can't really make any management decisions about the soybeans you're going to plant in that field that day. But yeah, it's going to make the most sense as far as management goes to sample in a cornfield after harvest before soybean planting. All right, I'm going to guess that's it. Once again, thank you for being here, and we hope to see you at the rest of our virtual field days. Thank you.